Hello, welcome to another edition of a CNA's Opus. My name is Rich. I'm a certified nursing assistant. The first thing that I want to start with is that Etherogogo-1's role in cancer is not a hypothesis. There's evidence for it, so there's evidence for this. It's been observed. The particular because Etherogogo-1 is not just one ion channel. It's just not one potassium ion channel. It's a family, and the one of particular interest is right here. It's KV10.1 within the Etherogogo-1 family, and that's expressed. I didn't say overexpressed. I said that's expressed in 75% of all cancers. And the reason that it's not overexpressed in cancers is because it's simply not expressed at all in people who don't have cancer. It's only expressed in the central nervous system. In people who do not have cancer, this particular potassium ion channel is only expressed in the central nervous system of people who do not have cancer. And people who do get cancer, in 75% of the cancers, Etherogogo-1, the KV10.1 subunits, is expressed in peripheral sites outside of the central nervous system. Right, so there's a variation there, which could be targeted. And one way that it could be targeted is through monoclonal antibodies. And the real true brilliance of this is that the monoclonal antibodies don't pass the blood-brain barrier. So the Etherogogo-1 KV10.1 subunits that are within the central nervous system are somewhat protected. They're protected, and the cancerous ones that are within the peripheral sites of the rest of the body, the monoclonal antibodies can attack. But the ones in the central nervous system, some of them are protected. So it's targeting cancer cells because it's being expressed in peripheral sites outside of the central nervous system on cancer cells, and then it can't get to some of the ones that are in the central nervous system because monoclonal antibodies can't pass the blood-brain barrier. So it offers that kind of protection right there. And that kind of treatment, which I hope to see very soon, is going to be it should do quite well. And how the Etherogogo-1 is increasing proliferation of cells and helping cancer along isn't quite known. All that is known is that it does. And another thing, one thing that is seen though, is that when Etherogogo-1 KV10.1 goes up in cells that are outside the central nervous system, is that something called vascular endothelial growth factor will go up. Now, vascular endothelial growth factor, particularly vascular endothelial growth factor A, is very important in angiogenesis. And since that's the part that's known, and angiogenesis is that pathway is very much known. In fact, it could go on. This could go on for weeks and weeks and weeks and weeks and weeks and weeks, just on angiogenesis. But I'm just going to quickly start with one. I just pulled some random things about angiogenesis here, and then I think I'm going to continue on with it. Angiogenesis here. So you have the Etherogogo-1 here, KV10.1, and then you have this kind of gap here because it seems to be sending signals down to another signaling pathway that's not quite understood. So there's a gap there that's increasing the expression of vascular endothelial growth factor A. And with vascular endothelial growth factor A, there's 8 different splice variants. So what you'll see up here is you have your introns and then your exons. And in the primary messenger RNA, you have both of them, and then the introns get cleaved out, and then the exons come together, and that's your mature messenger RNA that's going to be translated. So what is being there's an alternative splicing here. So you have exon 1, 2, 3, 4, 5, 6, and there's an alternative splicing at 6 and 7 here, which means at exon 6 and 7, it can cleave a little bit more out, or it can cleave a little bit less out. So you end up with a messenger RNA that's going to create a polypeptide that's either going to be longer or shorter in length. And that'll create your 8 different splice variants. And you'll put the splice variants, the number of amino acids in the polypeptide is a little subscript here. So you'll have vascular endothelial growth factor A 121 or 145 or 148 or 165 or 183 or 189 or 206 or 110. And that's the number of amino acids in the final polypeptide right there. The 110 here is a little bit unusual because it requires a second splicing to get to this 110. So the one that's 165 amino acids in length, or the one that's 189 amino acids in length, get a second splicing, which will come to a constant of 110 amino acids in length. So that will make up the vascular endothelial growth factor A protein, but the vascular endothelial growth factor A has to dimerize before it could actually work. And they're homodimers, which means they're the same. So vascular endothelial growth factor 121 can only dimerize with vascular endothelial growth factor 121. It can't dimerize with VEGF 110 or VEGF 189. They have to be the same to dimerize together. And they have to dimerize. The importance of dimerization is important later on, but they have to form a dimer. They have the protein's made, and then they have to come together. So the reason that they have to dimerize, and I have other things written down here, but I'm going off on this direction now. The reason that they have to dimerize is the vascular endothelial growth factor A is then released from the cell, and it'll go over to a capillary, and that'll kind of form a sprout. It'll start coming out towards the angiogenic center. But it binds to vascular endothelial growth factor A binds to a receptor tyrosine kinase, which I've gone over in my previous videos. And this particular receptor tyrosine kinase is vascular endothelial growth factor receptor 2. So vascular endothelial growth factor A binds with a receptor tyrosine kinase, which is vascular endothelial growth factor receptor 2. And again, receptor tyrosine kinases have a ligand binding domain. But the ligand binding domain of vascular endothelial growth factor receptor 2, the ligand binding domains are the same. There are 7 immunoglobulin domains within it. So that they're like the same thing. There's 7 immunoglobulin subdomains within the ligand binding region. And then over here, there's 7 immunoglobulin subunits within this ligand binding domain. So they're the same. So when the ligand comes here, which is vascular endothelial growth factor A, and the ligand comes here, which is same thing, there'd be nothing to, the same thing, so they're not coming together. It's not the ligand binding domain that forms that dimerization. It's actually the vascular endothelial growth factor A, the actual ligand that dimerizes them. Because they're the same on both sides, the vascular endothelial growth factor A, which is a dimer in of itself, comes in and brings the 2 ligand binding domains together. And on the other side, the intracellular fluid in the cytoplasm of the cell, that has the its tyrosine kinase domain is split. So there's a kinase insert there. So when it comes together and is activated, the 2 split regions will loop up and dimerize together. But the kinase insert domain isn't completely and entirely useless because it has tyrosine residues on it, which can be phosphorylated during the autophosphorylation process and the recruitment of and activation of certain proteins. But I want to hop back in here on the something that's the transcription factor for vascular endothelial growth factor A is a dimerization of hypoxia-inducible factor 1 alpha and hypoxia-inducible factor 1 beta, and they come together and they form the transcription factor for vascular endothelial growth factor A. So these 2 will go to the promoter region and they'll increase the affinity of RNA complex 2 to the promoter region, and then the transcription of vascular endothelial growth factor A will occur, and then it'll make the messenger RNA, which is spliced, and that will make the protein of vascular endothelial growth factor A, and then they'll dimerize and go off the cell to the capillaries and start doing that. So, but here the HIF1 alpha, or hypoxia-inducible factor 1 alpha, is constantly being made and constantly being destroyed. It's kind of like in previous videos you heard me talk about P53. Same kind of idea is that it's constantly being made and then constantly being destroyed, because it only needs to be there when the cell is hypoxic or low in oxygen. So what I'm going to just go over really quick is when the cell is in normoxic conditions, which means there's enough oxygen for the cell to survive. And here's the HIF1 alpha right here, and there's a specific region in here in the HIF1 alpha that contains 2 prolines, and the proline is this unusual amino acid that doesn't have the hydrogen off the amino group right here, because the amino group is part of this ring structure here that I usually don't put the hydrogens here, but it's important to put the hydrogen links off of the carbons right there, because I just want to show exactly what happens here. This particular enzyme, propyl hydroxylase enzyme, that comes over and takes off one of the hydrogens off the 4th carbon. You'll see it here, 1, 2, 3, 4. That'll take the hydrogen off the 4th carbon, and that'll add a hydroxyl group to it right there. So you'll see the HL right there. That'll add it, and that'll do it to both of the prolines. That'll do it to both the prolines. Then something called the von Hippel-Lindau protein, which is a tumor suppressor protein, will come down and bind to the hydroxylized prolines, or 4-hydroxylproline is what you would call the end product of that, because it's off the 4th carbon. So 4-hydroxylproline is the end product, and the von Hippel-Lindau tumor suppressant protein will come down and bind to it now, because it has that hydroxyl group on it right there. So that'll bind to it, and that'll add a ubiquitin molecule onto it, and the ubiquitin molecule will attach to the proteasome. It'll take it down to the proteasome, and it'll basically chop up the HIF1 alpha into smaller amino acids. Oh, polyubiquitin tail. I'm sorry. So it'll form like a polyubiquitin tail or a polyubiquitin chain, and that'll pull it into the proteasome and chop it up into smaller amino acid groups. And I don't think there's anything else I wanted to say. I'm kind of tired right now, but I think that's all I'm going to say right now. That's when it's under normoxic conditions. There's an aspartate protein residue right here, which is different, and that's when it's under hypoxic conditions, or oxygen levels are low, and that will prevent ubiquitination of HIF1 alpha right here. So it can bind with HIF1 beta and bind to the promoter region for vascular endothelial growth factor A, and it'll create the messenger RNA for it, and then the protein for it, which will again vascular endothelial growth factor A forms a homodimer. They have to come together at the same amino acid length. And I think that's it. Oh yeah, and the HIF1 beta here, or the hypoxia inducible factor 1 beta, that's being made constantly too, but it's useless until it dimerizes with HIF1 alpha. It can't do anything. So that's it for right now, and then I'll go over the hypoxic, what happens under hypoxic conditions, and so on and so forth. But the huge thing here is the role, the monoclonal antibodies in this, and the role of etherogogo-1 KB10.1 subunits in cancer, and that's going to be a huge treatment soon, I think, anyway. I don't know. So that's it. Rich, CNA. See you later.